everyone. We are now broadcasting. Thank you for attending the IMG webinar series and welcome. A few words about us who are conducting the webinar. My name is Natalia Ivanova. I am a scientist at the DOE Joint Genome Institute. And with me today answering your questions are Rekha Seshadri, Amy Chen, Ken Chu, Neha Varghese, and a few other people who are also scientists and software developers at the JGI. They will be answering your questions via chat. A few words about the format of this webinar. First of all, it will be recorded and it will be posted on the JGI YouTube channel. Your microphones will be muted and you won't be able to unmute yourself. However, you can ask the questions using Zoom chat. We will try to answer as many of them as possible during the webinar live. If we don't manage to get to all of them, or if we are not quite sure about the answer, then we will add the answers to the chat transcript. And this will be posted online together with the webinar recording. So even if your question was answered live during the webinar, we still encourage you to peruse this transcript because sometimes we can expand and you can get a better, more complete, or more comprehensive, more detailed answer. So that's kind of a few words about the webinar itself. Eventually, all of the recordings of these webinars, they will be posted on the website, the same website where you which you used to register, and so you can go back and review the slides and review the presentations and the tutorial. So the tutorial today will consist of two parts. First, I will go through a few introductory slides, and then I will do the IMG live demo. And because it is live demo, meaning that I will be clicking on links and doing analysis live in real time, sometimes depending on the network connectivity, speed, and everything else, it may be faster or it may be slower. So because it is live, we planned and we rehearsed this webinar for 1 hour, but if we run over a little bit, we apologize and we ask for your patience. In addition, after I'm gone through the slides, I will pause and my colleagues, they will review the questions, and if there are a few very common ones, they will broadcast, we will answer them live on air. And the same story with the I will have the breaks while I'm doing the live demo of IMG. So next slide is, I have to say a few words about who we are. We represent the DOE Joint Genome Institute, which is a US Department of Energy user facility. Department of Energy Office of Science has these user facilities that enable large-scale science relevant to the DOE mission areas, such as clean energy, bioremediation, environmental cleanup, and other environmental challenges. So DOE JGI Joint Genome Institute is a facility which promotes omics research, essentially sequencing, providing sequencing capacity to the scientists worldwide in order to characterize the genomes, metagenomes, transcriptomes, metatranscriptomes of plant, fungi, bacteria, and archaea, and environmental communities of all of them. We are funded, as I said, we are funded by the DOE Office of Science. We were opened in 1998. Currently, we have about 280 people. We are located in Berkeley, California. And if you want more details about the JGI, as well as about the sequencing programs, community science programs, and other programs that we have, please visit our website, which is this link, jgidoe.gov. Today, we will be using the system which is called IMG, and specifically, I will be using the secure system which is called IMG MER. IMG exists in multiple flavors. One of them is this IMGM, which is completely public and open to everybody. It doesn't require login information, but it has limited capabilities. The one that we will be focusing on is IMG MER, and this one requires that you get an account with IMG and that you log into IMG. And you can figure out that you are logged into IMG if you have your username in the upper right part of your screen, and you have this logout link. In addition, you would have this workspace option in the menu. So other IMG flavors include IMG ABC and IMG VR. These are the systems that are dedicated specifically to the data about biosynthetic gene clusters and viruses, respectively. So other capabilities of IMG MER is that you can submit your own data, and IMG will be annotating this data for you. You can also annotate your own data sets, the results, access workspace functionality, as well as this important part about submitting computation jobs that are much larger computations that cannot be performed on the fly and have to be run in the background. So if you have difficulty getting an account with IMG, or you have an account and you have difficulty logging into IMG, there is this help option in the rightmost corner of the main IMG menu. If you click on it, there will be a section called Contact Us and the Contact Us link. If you click on it, you can submit a ticket so that we could address your problems. So now we will be talking about IMG carts and workspaces. So, and these are essentially the IMG's shopping cart. I hope that now with the quarantine and everything else, everybody is familiar with Amazon and the idea of having a cart where you add the items that you are interested in that you intend to purchase later on. And this is a temporary cart, meaning that you log out of the system and then the items in your cart disappear. But in addition to this cart, Amazon also have this saved list, like this saved for later. Meaning that you are not going to buy them right now, but you are saving them for the future purchase. IMG is organized exactly the same way, except that instead of one cart, it has multiple carts. And these carts and their contents are listed here above the main IMG menu. So IMG has genome cart, scaffold cart, function cart, gene cart. And this is because IMG has a much better defined set of objects than Amazon, where you can buy essentially anything. So in addition, IMG has this workspace, which is sort of equivalent to Amazon's saved for later list. So carts are equivalent to Amazon cart, and workspace is equivalent to saved for later, meaning that there are these sets of genes, genomes, scaffolds, and functions that you can save and go back again and again for analysis, export, and so on. So let's say a few words about the genome cart and workspace, because it is functioning slightly differently from the rest of the carts and workspaces on IMG. So there is genome cart, as you have seen, and there is this genome set workspace. I apologize, it should be genome set. So both of them have export capabilities. And by export, IMG means that you can customize the data that you want exported. You can select the specific data sets for which you want to export the metadata, and you can select the columns in your export. The metadata will be exported as tab delimited file, which can be opened in Excel. In addition, both have download links, which is not done by IMG, but instead it links out to the JGI data portal. So Genome Workspace has these three analysis tools. One is called Function Profile, and we will be focusing extensively on this one today. The other one is BLAST. This was covered in the previous webinar. And the third one is Statistical Analysis tool, which will be covered in the next webinar. So Genome Card does not have any analysis tools because it has the analysis tools for genomes are in this Compare Genomes menu. In addition, Genome Card serves as a filter in many other tools, meaning that in many cases, IMG does not let you run the search or analysis on the entire content of IMG simply because it is too large. In this case, it asks you to select a subset of data sets. And by default, in most cases, the content of your genome card will be the default set, the filter for all of these analysis tools. Now about gene cart and workspace. So again, the same story, there is the cart and the workspace, and they have similar capability in a sense that you can export their content, both the metadata and the sequence. in both cases, the sequence could be nucleotide sequence or protein sequence for the protein coding genes. Both have analysis tools, and they are different for the gene card and gene set workspace. So in the workspace, there is the function profile, while in gene card, there is an option for multiple sequence alignment of the gene sequences and also the chromosomal neighborhood view. Again, the export in this case means that you can select the genes, that you can select the type of the sequence that you want to export and the types of the metadata. However, there is a limited kind of scope in this case because in gene card, for instance, it allows you to store only up to 20,000 genes. So all of this was covered extensively in this data download and export webinar, which you can view if you go to the webinar registration page. The scaffold card and workspace again is similar to gene card. So it allows you to export metadata or sequence. In this case, the sequence is only nucleotide sequence of the scaffolds and context. And both have analysis tools. Analysis tools in scaffold card include histograms of GC content and read depth, as well as k-mer composition tool. The scaffold set workspace includes function profile tool, which we'll be again covering today extensively, as well as k-mer composition tool and file distribution, which shows you the distribution of the best hits of the proteins encoded on the scaffold against the isolate genomes in IMG. So these tools, we won't be able to cover all of them, obviously, including this k-mer composition, and that's why you were sent the document, the sort of like the what we call IMG tips and tricks, which has the details of k-mer composition tool, as well as the set sharing capabilities. We encourage you to review this document if you are interested in either of them. The topic today, basically the biological system of today's demo, is the same as of the previous webinar, and it is Bradyrhizobium bacterium in the environment. Bradyrhizobium is a genus of alpha proteobacteria, and these are root nodulating bacteria, meaning that they are symbionts of legumes that fix nitrogen and supply it to the plant host. And it is a remarkable system in a sense that it involves a lot of interactions, complex interactions between the host plant and the symbiont. So the plant starts the interaction by secreting the flavonoids, which Bradyrhizobium recognizes as a signal that there is a potential host plant in the environment. In response, Bradyrhizobium secretes so-called nod factor, which is a compound produced by a set of nodulation genes usually encoded on this symbiotic plasmid. So this nod factor is recognized by the host plant as that there is a potential symbiotic bacterium in the environment. And the host plant, it sort of turns off its innate immune system, which will normally try to kill any invading bacteria. In this case, instead of killing the Bradyrhizobium, it actually creates a specific environment called nodule, where the bacteria multiply, and also the plant creates the environment which is conducive to nitrogen fixation because it is essentially anaerobic. In addition, the host plant supplies the bacterium with carbon source, while the bacterium provides nitrogen to the plant. And it is a very interesting system. And again, you are encouraged to read this review if you are interested in the details. So the research questions that we will be answering today, they are kind of an extension and build upon the two previous IMG webinars. And one of them is the previous one, the IMG sequence similarity search, which discussed the type strains of bacteria and archaea as anchor strains, meaning that this is the first experimentally characterized strain of the lineage, and it is sort of defines the characteristic of the lineage. So the second webinar is about this metagenome beams. It was in the first IMG webinar series where we told you what are the metagenome beams in IMG, how they were generated, as well as what are the tools to access these metagenome beams. And we will use this metagenome beams for Bradyrhizobium as environmental representatives of Bradyrhizobium genus. The specific questions, research questions that we will be answering are, the first one is, are nitrogen fixation and nodulation common features of all Bradyrhizobium type strains? Or is it not kind of a mandatory characteristic of Bradyrhizobium genus? So the second research question is what about the metagenome beams as environmental representatives of bradyrhizobium? Do all of them have nitrogen fixation and modulation genes, or are they absent? The third question is, and it relates to these metagenome beams, in case the nitrogen fixation and modulation genes are absent from the beams, do the remainder of metagenomes, do the entire metagenomes in which these bradyrhizobium beams were found, do they have this nitrogen fixation and modulation genes? And the last slide is the outline of the demo itself. This is how we will proceed to answer these particular research questions. So in order to answer the first question about the presence and absence of nitrogen fixation and modulation in bradyrhizobium type strains, we will be using KEGG orthology terms. And KEGG stands for Kyoto Encyclopedia of Genes and Genomes, and it is a very popular collection of metabolic pathways mainly, but they also have their own annotation system for protein coding genes, which is called this KEGG orthology. So we will be using this KEGG orthology annotation system to find nitrogen fixation and modulation terms, KO terms, and then we will view the profile. We will use function card to view the profile of the genomes of type strains of bradyrhizobium against these KEGG orthology terms for modulation and nitrogen fixation. In order to answer the second question about metagenome beams, first we will create a set of these KO terms for nitrogen fixation and modulation. Then we will find and copy metagenome beams with the taxonomy of bradyrhizobium and high degree of completeness. We will be looking only at nearly complete beams of with completeness above at least 98%. And we will view profile of these beams against, again, this set of KO terms using the scaffold set workspace. In order to answer the last question, we will create a set of metagenomes in which these bradyrhizobium beams were found. And then we will view the profile of metagenome set against the function set of KEGG orthology terms. And so this is the end of this introductory slides, and I will pause and ask if there are any questions that I need to answer immediately.
No, Natalia.
Excellent. Then we will proceed with the IMG demo. So let me get rid of the slides. Can you see my screen?
Yeah.
Okay, excellent. So now we will proceed with live demo in IMG. This is the home page of IMG MER. And as I said, we will be using today IMG MER and not IMG, the publicly available system, because we will be using the functionality of the workspace, which is found here. And as we discussed in the introduction, I can figure out that I am in IMG MER because I have my username on top of the screen, logout link, workspace menu. And if you have difficulty obtaining the IMG account, or if you have difficulty logging into your IMG account, then you can go to this help menu. I will open it in a new tab to show you. So where there is this contact us and other section and this contact us link, which will create a ticket for us to address your problem. Now let's go back and let's try to figure out how we can answer the first question about how we can find the type strains of the bradyrhizobium genome. So the general answer is that we will go to this find genome search. And I'm not going to run the query, and I'm not going to discuss the details of how you construct this query because this was covered in multiple webinars. I will only point you to this list of public queries. If you go to this, click on this public list of queries that were saved for you, the query number 4 here is the query that was used to retrieve the type strains of bradyrhizobium genomes. And if you select it and click on Add Selected to History Card, this query will be reconstructed. After you run this query, you will get a set of 23 genomes, and you have to select all of them and click on Add Selected to Genome Card. And that's what I have right now in my genome card, 23 genomes. I can click on this link on top of, above the IMG menu, which lists the content of cards. And if I click on it, I will see the list of these 23 genomes, and these are bradyrhizobium type strains. So the next thing is that now that I have my type strains of bradyrhizobium on my genome card, the next question is, how do I find these tag orthology terms that characterize nitrogen fixation and nodulation? Again, IMG has multiple ways of how we can do it. I will show you today one of them. So in Find Functions menu, there is this option of tag. And in tag, there are multiple ways. There is just flat list of tag orthology terms. Then there is the tag module list, and on the bottom of the menu, there is the pathways and orthology, which is the pathways is what most people know about tag. These are the maps that show the compounds and the enzymes catalyzing the reactions between these compounds and so on. But that's not what I am going to use today. Instead, I am going to use this tag module list. The reason is because in many tag pathways, most actually tag pathways, these tag maps, they cover very large areas of metabolism. So there is a map called nitrogen metabolism, which includes all kinds of things, including denitrification, nitrogen fixation, and many other areas. Tag modules are much smaller, much more granular sets of tag orthology terms. And that's why I'm going to use them today. So let's open it in a new tab. So here I have the page where IMG is opening for me the entire list of these tag modules. And it has right now 731 of them. Now this is the typical IMG table, which I can filter, sort, and so on and so on. And it lists several things. First of all, it is the usual thing, the module ID, then the name of the module, and this is where you would expect to find the terms such as nitrogen fixation and nodulation. The next column shows you the module type, because these correspond to several different things. The module type complex means that it is a protein complex. In this case, this top row, it is actually an ABC transporter, and it is a protein complex. So the next one is the definition, and it is essentially a kind of a logical expression that tells you that in order for this module to exist, you have to have these 4 tag orthology terms. Other types of the tag modules are pathway, like this one, or the next one, and also this kind of systematic function sets, meaning that these are the functions that are likely to participate in this activity, and this is, for instance, two-component regulatory system, but it is not necessarily a protein complex, and it has a lot of pathway and so on. So let's filter this table by searching for nitrogen as a term. And again, so basically I'm doing this table filtering. I am just typing the keyword nitrogen, and then I am clicking on apply. Again, this was reviewed in multiple IMG webinars. We encourage you to review some or all of them. There are 5 different tag modules that have nitrogen in their module names. And we are interested in this particular one, nitrogen fixation, which starts from nitrogen and goes to ammonia, and this is a pathway. So again, I will click on this module ID, and I will open it in a new tab. So this particular module, again, the module details page, the usual IMG details page, which IMG has for most objects that it has, it lists on the bottom this set of tag orthology, and these are specific annotations that are being assigned to the protein coding genes in IMG, and that's how we know that this protein in a specific genome are connected to this tag module of nitrogen fixation. Let's select all of them. And you have 2 options here. You can either add them to the function card, or you can save them to the workspace. Let's start with adding them to the function card. So here is my function card, and these are the TO terms that are now added to my function card. Let's close this tab, and let's go back to the tab with the list of tag modules. So we found the genes for the nitrogen fixation. We found the, I'm sorry, not the genes, the tag orthology terms for nitrogen fixation. Now let's do the same for nodulation. Again, I am filtering this table by typing nodule and clicking on apply. There is exactly one module called modulation, and in this case, it is signature. It is, again, it is not pathway, meaning that they don't know quite how the genes, how the TO terms in these particular modules are connected to each other. Again, let's click on it and open it in a new tab. So in this case, again, the same story, the tag module details page and the table on the bottom, which lists the specific tag orthology terms that are assigned to this tag module. So I can select all of them and also add them to the function card. But now I notice that there is this term called NOT-D, which is a transcriptional regulator. And I will probably deselect it. And the reason for that is that generally the annotations of transcriptional regulators with specific activity, such as in this case, that it is modulation specific transcription factor, that they are notoriously unreliable. So I won't use this particular tag orthology term. And I will click again on the Add Selected to Function card. So now I have 7 functions in my function card. The first batch, these are tag orthology terms for nitrogen fixation, and the second batch, the tag orthology terms that are specific for nodulation. Now, in order to see whether these functions are present in my set of genomes, first of all, I will click on Select All in order to select all of them. And the second thing that I will do is I will go to this Profile and Alignment tab, which allows you to run this profile function of IMG, and we will go further down to see what it does. So remember in the introductory slide, I told you that the content of your genome card is used as a filter wherever IMG asks you to specify the set of genomes or metagenomes on which you want to run the analysis. Here it is, right? So basically all of the genomes from my genome card, regardless of their sequencing status, and there are 23 of them, and they're selected by default. As you can see, this option of function profile, right? So basically it has limits why we are kind of adding the genome card and so on and so on. Because you can select only up to 500 genomes. And it is much more convenient if you have the kind of the smaller set than that, and then you have the genomes of interest on your genome card. Now it has 2 options. You can view functions versus genomes, or you can view genomes versus functions. The difference is that in the first case, functions will be displayed as rows, and in the second case, genomes will be displayed as rows. In my case, I have more genomes than functions, so I would want genomes on the rows and functions as columns, so I will click on this View Genomes versus Functions. So this will generate for me a function profile, what IMG is calling function profile, which is nothing but a fancy word for a matrix, where the genomes are displayed on the rows, like this Bradyrhizobium arachidis, and the tag orthology terms or any other function that you would have on your function card will be displayed as columns. If you mouse over on the column name, it tells you the name. What is this particular tag orthology term? What does it stand for? So this one, for instance, is NIF-H protein that many people are using as a signature for nitrogen fixation process. And in the cells of this matrix, you have the counts of annotations with this particular tag orthology term, the alpha subunit of nitrogenase, found in this particular genome, Bradyrhizobium arachidis, and so on and so on. And if you mouse over, you can see that this is linked to something. If you click on it, you will be eventually end up at the gene details page of this particular gene. The gene annotated with this tag orthology function found in this particular genome. In addition to looking at these genes one by one, there is this option on the bottom which shows you all genes in this particular matrix. So just from looking at this table, what we can say? We can say that there is this weird tag orthology term which is not found in any of our Bradyrhizobia. And if you go to tag and if you look at what this function is, it is actually an alternative nitrogenase which is found only in Azotobacter species, which is not a member of this Bradyrhizobium genus. So we do not expect to find it in Bradyrhizobium genus, in the genomes from Bradyrhizobium genus. The rest of the KO terms, both for nitrogenase and for nodulation, they're actually found in all genomes in my set, in all type strains, except for this one, bradyrhizobium symbiodeficient, which has zeros for all nitrogen fixation and all modulation genes. If you click on this name of this genome, you will get to the genome details page of it. And from there, you can navigate to the NCBI, and there you can find publications about this genome. And you can find out that it is true, what IMG is showing you is absolutely true, that people who isolated this particular type strain, that they characterized it experimentally, and they found out that it does not nodulate, it does not fix nitrogen, even though it is found, it was isolated from the rhizosphere of soybean. So, and this ends the first research question that we had, whether the nodulation and nitrogen fixation genes are found in all type strains of bradyrhizobium and therefore are the feature of the entire bradyrhizobium genus. The answer to this question is no. We have at least one type strain of bradyrhizobium which does not have nitrogen fixation and nodulation genes. I will stop here and I will ask if there are any questions that I need to answer immediately.
No, Natalia.
Excellent. So now let's proceed with the second question, which is now that we looked at the type strains, now that we looked at the bradyrhizobium genomes of the strains that grow in isolate culture, and they're used as an anchor sort of to characterize all the rest of the bradyrhizobium in the environment. What about the truly environmental bradyrhizobia that we find in metagenomes and that we characterize as metagenome beams? What about them? Do all of them have nitrogen fixation genes? Do all of them have nodulation genes? How do we basically, how do we approach this question? So first of all, let's go and let's try to find these bradyrhizobium genes, bradyrhizobium beams in the metagenomes. Again, there was a webinar about metagenome beams, which discussed about how you can find them. So in this find genomes menu, there is metagenome beams and beam search menu. I won't run the search, but I will tell you that the same way as we have this public list of queries for genome search, we have similar list of public queries in this public list in metagenome beam search. If you click on this public list, you will be redirected to this public list of saved queries, and the query that I would be using is the second one, with the DTTBTK genus bradyrhizobium and beam statistics, the completeness of at least 98%. So if you can select this query, you can click on Add Selected to History card. It will be added to your search history, metagenome beam search history, and after that you can rerun this query, and you will get 12 metagenome beams that have this genus of bradyrhizobium and that have the completeness statistics of at least 98%. So I did exactly that. And I basically, after that, you can select all of them, and you will have an option of saving them all to your workspace. And in this case, they will be saved to your scaffold set workspace, not genome, because these are metagenome beams and not isolate genomes. So if I go to my workspace scaffold sets now, I will have the, again, I'm an active IMG user, so I will have a lot of beams, different beams, including this set of bradyrhizobium beams that have at least 98% completeness. In order to make it slightly easier for everyone to see, I will filter this table so that only these beams show up. And I will say that I'm basically selecting only the ones that have this scaffold set in the name. So this is the set of 12 beams which have bradyrhizobium genus and at least 98% completeness. In the scaffold sets, the default things that you can see is the number of scaffolds assigned to each of these beams. And you can see that they're kind of variable quality, because the beam with the smallest number of scaffolds has only 3 of them, and the beam with the largest number of scaffolds is actually an order of magnitude more with 324 scaffolds. This means, this tells me that this beam, with 33 scaffolds only, it is very well defined with very well assembled beam, while this one with 324 is kind of suspicious because it is probably highly fragmented, as many of the scaffolds in this beam are very, very short. So now that we have these beams, saved in our scaffold workspace. In order to see whether they have or they don't have this nitrogen fixation and nodulation genes, I would select all of them by clicking on Select Page, and then I will use this Function Profile tab in the scaffold card. So this scaffold card, basically this Function Profile option, is very similar to what you have seen in the function card, except that it allows you to do two things. First, it allows you to select a specific set of functions that you want to see. And we will go in a moment about how you create this set of functions that you want to see. The second option is this one: use all functions of a certain type. So such as this COG, KEG, and so on and so on. PFAMs and Tiger FAMs. So for this exercise, I'm interested only in nodulation and nitrogen fixation genes. So I will be using these functions, only functions in a set. And this is actually the recommended way of using IMG because, first of all, the larger these sets of functions, all functions of type, they're fairly large. So for instance, this KEG orthology and PFAM, they have each approximately 25,000 functions. And if you look here, it tells you that the result is limited. So the total number of cells in your search cannot be more than 100,000. So you can imagine if you are selecting 25,000 PFAMs, you cannot view the profiles for more than 4 metagenome beams if you are selecting only counts per set. And that's why we encourage you to download, if you are interested in all COGs, in all PFAMs, in all KEG orthology terms, please go and download the data from the JGI portal. And please use this tool only for the select set of functions, only for specific set of functions. Now let's go and look how we can create this set of functions that we are interested in. Well, we can go back to our function card, which is again, my analysis cards are here. And this shows me that it still has these 7 functions in my card. Let's open it in a new menu, in a new tab. All of them are selected. And here in function card, I will go to this Upload and Export and Save tab, which allows me to save a function set to my workspace by providing a name. And in this case, basically, I already tried it, and this is the name that I would select: nitrogen fixation nodulation. So, and then after I click Save Selected to Workspace, I will be redirected to my function set workspace, where now I have this 7 functions set saved to this particular set. And again, I'm an active IMG user, so I have a lot of different function sets for COGs, PFAMs, KO terms, and so on and so on. So what we want in this case, we want to go back to our scaffold set workspace, and we want to select again these 12 sets from the bradyrhizobium metagenome beams that are at least 98% complete. And we want to go to this Function Profile tab. Now that I have this set of functions for nodulation and nitrogen fixation, all I need to do is I need to find it here. And here it is, nitrogen fixation nodulation. If I select it, I have again 3 options. I can do the counts per set, counts per scaffold, or I can do both. In this case, I'm not interested which scaffolds, specific scaffolds have this particular gene, so I will select the counts per set, meaning that it will show me the total number of of this particular function found in the entire scaffold set, in the entire beam. And as you can see, there is the option here for submitting a computational job using messaging system. If you are selecting not 12 beams, but you are selecting hundreds of beams, or you are selecting a lot more functions than this set of 7 that I have here, you are encouraged to submit a job, meaning that the computation will run in the background, and then you will receive an email notifying you that your job is complete, and it will provide a link. And you can also find the results of your completed jobs here. If you go to Workspace, My Jobs, that's where the list of your completed jobs and also the job status will be found. So I am selecting a small set of beans and a small set of functions, so I expect it to run in real time. So I will click on this scaffold set function profile. And in order to save time, basically, while it is running, I can tell you that this is what the results will look like after it finishes. So it will bring you the table with 2 tabs. Unlike the presentation on the function card, it will show you 2 tabs. And it will separate the functions which have non-zero counts in at least some scaffold sets from another tab which has only the functions that have 0 counts, that are not found in any of the scaffold sets that we have selected. Here it is. It's the same, basically, the same results. Now it brought the results. So let's go to this tab with functions with 0 count. What do we see here? First, we see this nitrogenase delta subunit, which is we didn't see in any of the type strains of bradyrhizobium, and which is the azotobacter alternative nitrogenase, which is not expected to be found in bradyrhizobium. But in addition, we see all 3 nodulation-related functions that we had in our set. So we can immediately say that we did not find any nodulation genes in any of these high completeness beans, bradyrhizobium beans that we found in the metagenome. So now let's go back and look at the functions with counts. Even these ones, as you can see, the first beans in this set have 0 counts, and only further down you start having 1 of each and further on. So just like in the function card function profile, you have this option of selecting, of clicking on the gene count, or you can select all of the genes, for instance, that were assigned to this particular function by selecting this KO term, and then you can add the genes of this selected function to the gene card, or you can save them as a gene set. The same thing you can do either adding the selected functions to the function card or saving them to the as a function set to the workspace. So, and this completes the exercise of the answer to the question number 2. Do the environmental representatives of bradyrhizobium, do the metagenome beams used as environmental representatives of bradyrhizobium, do they have the nodulation and nitrogen fixation genes the same way the types, nearly all type strains of bradyrhizobium have? And the answer is no. Actually, none of them, for some reason, have any nodulation genes. And very few of them actually have nitrogen fixation genes as well. So this concludes the second part of the live demo. And again, I will ask Rekha if there are any questions that I need to answer immediately.
No.
Excellent. So let's proceed with the third part of the IMG demo. First, it's a little bit it requires a little bit of explanation. So we didn't find the nitrogen fixation or nodulation genes. We actually didn't find nodulation genes in any of this high completeness. Again, we were looking for the genes with at least 98% completeness, right? The beans. These are high completeness beans, and we didn't find nodulation genes in any of them. And we found nitrogenase genes in very few of them. Why could that be? Well, there could be 2 explanations. The first one is, as usual, it is biological explanation. Somehow our characterization of bradyrhizobium is biased, and we have basically bradyrhizobium from specific environments, these type strains, and that's why all of them have this nitrogen fixation and nodulation genes. Essentially, people were looking for bradyrhizobium that is nodulating specifically, and that's why they found all of the type strains that have nodulation and nitrogen fixation. And they didn't bother to isolate bradyrhizobium from other environments where they don't have nitrogen fixation or nodulation genes. So that's one of the, and here in the real in the wild, in the environmental bradyrhizobium, we see the real picture, the real functional content of these particular genes. So that's one explanation. But there is also an alternative explanation. And it is the explanation that the absence of nodulation and nitrogen fixation genes is a technical artifact of this metagenome beaming process that we have. And it's actually a very likely explanation if you think about how it is done. So if you attended this Metagenome Beams webinar, or if you watched it, the recording of it, you would know that metagenome beaming is using 2 parameters. in order to assign metagenome context to the same beam, to the same set of contexts and scaffolds that are likely coming from the same population. It is using first the oligonucleotide composition of this context, because it is kind of well established that for multiple reasons, the oligonucleotide composition of the genome is relatively consistent. So the second parameter that it is using, the metagenome binning, it is read depth, or the coverage of this context. And again, the contexts that are coming from the same genome, they're expected to have approximately the same coverage. So these are the two parameters. Now, let's go back to the introductory slides, which were saying that nodulation and nitrogen fixation genes are often found not on the chromosome, but they are found on so-called symbiotic plasmids. And let's think about how this will affect the metagenome binning that we perform on all of these metagenomic data sets. And if you think about it, it will seem more and more likely that binning will fail when we have a strain, when we have a population that has plasmids. Why would that be? Well, first of all, plasmids often have different oligonucleotide composition from the rest of the genome, from the chromosome. In many cases, plasmids have lower GC content than the chromosomes of the genome. So that's one thing. So they will have different oligonucleotide composition, and they won't look like the chromosome of the genome. On the other hand, plasmids often have more copies, are present in more copies than the chromosome of the genome. So this will fail the second criterion used for binning, the read depth or coverage, because again, the plasmid for the binning tool that we are using, the plasmid will look differently than the chromosome of the genome. So it is highly likely that the plasmid genes won't be included in the metagenome beam, simply because the plasmid of the strain does not look like anything that, like the chromosome of the same strain. So that's kind of the long and winding explanation about why we are going to do the third exercise. So we want to figure out whether it is true that the metagenome beams that we have, that they are an honest representation of the environmental bradyrhizobium strains, or whether they lack this nitrogen fixation and nodulation genes because of the failure of this binning process that does not assign context that originates from plasmids to the metagenome beam, which mostly represents the chromosome of the strain. So how can we figure out whether it is the biological phenomenon or whether it is the failure of the binning? So in this case, we will do again the same functional profile exercise, but we will do it on the entire metagenome, not only on these bradyrhizobium beams from this metagenome. So let's go back to this workspace, do the scaffold sets. And let's select these beams, bradyrhizobium beams that we have here. And after that, we will use another tab in the scaffold card, which is called Genomes and Genes. And this is essentially the tab that allows you to navigate between different sets. So it allows you to select this set of scaffolds and save all of the genes encoded on this set of scaffolds as a gene set. It also allows you to save the genomes in which these scaffold sets were found as a genome set. And this is exactly what we are going to do. We will save the genomes in which the metagenomes specifically in which these beams were found as a metagenome set. And we will call this metagenome set, again, we have to provide a name, and we will call it metagenomes with Brady marks, just for the fun of it. And we will save them to my genome workspace now. If I click on this Save Genomes of Selected Scaffold Sets, it will create for me so first of all, I already have it, and that's why it tells me that it already exists, and I have to provide a non-redundant name. So let's go to the genome sets, and let's review. Basically, because I already have saved it here, let's go and find it. So this is metagenomes with Brady marks set, It is 12 of them, basically 1 meg per metagenome, and we can click on it, and we can see what are these metagenomes with their names and so on. So, and you can see that there is some bradyrhizobial megs nearly complete from marine environment. Then there are some soil communities. Then there are the fungus garden communities, fracking water. Then there is the rhizosphere from a plant from Puerto Rico, again soil, hot spring, Arctic peat, and also some root nodules. So the environments actually from which these bradyrhizobium beans were generated, they are very, very diverse. Now that we have them in this set, and we have all of the functions, I will remind you that we have all of the functions for the nodulation and nitrogen fixation as a function set. Now we can run the function profile for the metagenome set. It will show you the counts of this function, not in the set of scaffolds that were assigned to this particular mag, to this particular bean, metagenome bean, but it will show you the function assignments for the entire metagenome. Again, the same story, I select the genome set, the set of metagenomes. In this case, I go to this function profile tab, and I select the set with the functions for nodulation and nitrogen fixation. It is this set, nitrogen fixation, nodulation. In this case, basically, I want to see the counts per genome, because I want to see how many hits, how many counts of each particular cog is found in each metagenome. After that, I will click on this genome set function profile. And again, basically, you have seen on the bottom, let's go back. Again, I'm selecting a relatively small set of metagenomes, and I'm selecting a small set of functions, right? But if you would have a much larger set of metagenomes, and if you had a much larger set of functions, you would have to use this submit as computation job using message system. And again, you have to be mindful that the total number of cells in the table should be less than 100,000, because otherwise, basically, it is it's essentially basically it will simply time out. So again, the set, the counts per genome, genome set function profile. Again, IMG splits in this case the functions that have some counts and the tab which has functions with 0 counts. And now the only function that has 0 counts in these metagenomes is this weird nitrogenase, which is not expected to be found in bradyrhizobium species. All of the rest of the functions that we have, they all have counts in these metagenomes, even those that have 0 counts of nitrogenase, for instance, in the metagenome in bradyrhizobium bean. And many of them have these counts, some counts of proteins for nodulation proteins as well. So what does it look like? So for instance, let's look at this particular metagenome, and I can tell you immediately that basically that the bean from this metagenome didn't have any nodulation genes and it didn't have any nitrogen fixation genes. But we do have nitrogen fixation genes and we do have nodulation genes in the rest of the metagenome, obviously. So now the question is, it looks more and more like a technical failure, right? So basically that the metagenome binning pipeline did not put the context that have these nitrogen fixation genes and nodulation genes into the same bin of bradyrhizobia. If you want to figure out why that have happened, you can click on this gene count and open it in a new tab, and then go to the gene details page of this particular gene. If I click on this gene ID, I will be brought to the gene details page we have seen many times, and it shows you all of the details about this particular object, the gene, including this scaffold source, the scaffold on which this gene was predicted. And in parentheses here, it shows you the length of the scaffold. And it is actually a very, very short scaffold of only 1,000 nucleotides. If you scroll further down, it is indeed a very, very short scaffold which has only 2 genes, one nitrogenase subunit and another nitrogenase subunit. So, and I can immediately tell you that this is the reason why this scaffold wasn't assigned to this particular, to the bradyrhizobium bean. If we click on this scaffold name, it will give you the details about the scaffold, including the lineage. And the lineage of the scaffold is bradyrhizobium. So it is the scaffold which should have been assigned to the bin of bradyrhizobium. But it wasn't, because our binning pipeline has a strict cutoff for the length of the scaffold that it is trying to bin. Essentially, basically, we are not looking at any scaffolds that are that short, simply because binning of them will result in many false positives, and which essentially show up in the bins as contamination. And so that is the answer to the second question, to the third question of the demo. So we do have the nitrogen fixation and nodulation genes in the remainder of the metagenome, from which we extracted this bradyrhizobium high completeness metagenome beam. These genes that are for nitrogenase and nodulation genes, they were not assigned to the beams for multiple reasons. One of them is that many of them were found on the scaffolds that are too short to be binned. The second could be is that they may be, again, the plasmid genes and plasmid sequences, and they may have different oligonucleotide composition and different read depth coverage patterns than the rest of the genome. So, and it is so the answer is kind of twofold. But still, basically, I will point you to the two metagenomes, and both of them are aquatic metagenomes that don't have any nitrogen fixation and don't have any nodulation genes. And then another metagenome which also doesn't have any nitrogen fixation nodulation genes, despite coming from the soil community. So it looks like a mixed message. So some of the environmental bradyrhizobium do not have nitrogen fixation and nodulation genes. So our type strains are indeed biased somewhat towards the nodulating bradyrhizobium. But on the other hand, in many cases, metagenome binning sort of fails for the plasmid genes and for the genes that are found on the shorter context. So that concludes the third question of the demo. And again, I will ask if there are any immediate questions that I have to answer.
No, Natalia.
Okay, excellent. So then we are under the time, and I again thank you for participating in this IMG webinar. As I said, we will post the recording of the webinar on the JGI YouTube channel, and you will receive an email notification, and the chat transcript with the answers will be posted together with the recording. Thank you so much, and have a great day.